Hello students, a warm welcome to the virtual practical class on the development of the gastrointestinal system. For this practical class, we have captured the pictures of the embryology models available in the NDMC Medical College and Hindu Rao Hospital. At present, as you are not able to see those models, please see the pictures of the models and corroborate with the pictures in the Langman's textbook of embryology. Here, this picture is showing the sagittal midline section of the embryo at day 14. The gastrointestinal tract is the main organ system derived from the endodermal germ layer. The germ layer covers the ventral surface of the embryo and forms the roof of the yolk sac. With the development and growth of the brain vesicles, however, the embryonic disc begins to bulge into the amniotic cavity. Lengthening of the neural tube now causes the embryo to curve into fetal position as the head and tail regions moves ventrally. These two arrows shows the folding of the neural tube and the whole embryo. Simultaneously, two lateral folds of the body wall also form, which move ventrally to close the ventral body wall, which is not shown here. At, at its cephalic end, the foregut is temporarily bounded by an ectodermal endodermal junction or membrane called the oropharyngeal membrane. The hindgut also terminates temporarily at an ectodermal endodermal membrane called the cloacal membrane. Cephalic to the oropharyngeal membrane is the cardiac area where the heart tubes develop. Hindgut also terminates temporarily in an ectodermal endodermal membrane, the cloacal membrane. Caudal to the cloacal membrane, a diverticulum from the yolk sac forms the allantois. The function of the allantois are in detail has to be covered in the urogenital system development. It is a vestigial structure that serves as a respiratory and waste storage organ for the avian system. It extends from the hindgut region to the umbilical cord and forms the urachus, which will be covered detailed in the urogenital system. This is slightly advanced section of an embryo at day 17. As a result of continuous head fold, tail fold and the lateral body fold, a portion of the yolk sac gets incorporated anteriorly and posteriorly to form the future foregut and the hindgut. This is more advanced age of the embryo, that is at day 24. Note that growth of the neural tube cranially and caudally with the foldings. Therefore, with the more folding of the embryo, the more portion of the yolk sac is incorporated inside the embryo anteriorly and posteriorly to form the foregut and the hindgut respectively. The ventral body wall also closes completely except at the region of the umbilicus where the connecting stalk, the yolk sac and the its remnants remain attached. Failure of the lateral body wall folds results in the ventral body wall defect that we have done earlier. The midgut communicates with the yolk sac by way of a broad stalk, that is the vitalointestinal duct. This duct is wide initially, but with further growth of the embryo it becomes narrow and much longer. By the end of the fifth week, the yolk sac, the vitalointestinal duct, the allantois, and the umbilical vessels are restricted to the umbilical region. The role of the yolk sac uh, is not clear. It may function as a primitive nutritive organ during the earliest stages of development prior to the establishment of blood vessels. It also contributes some of the blood cells, although the role is very transitory. One of the main functions is to house the germ cells that reside in the posterior wall and later migrate to the developing gonads to form the egg and sperm. Hence, the endodermal germ layer initially forms the epithelial lining of the primitive gut tube, and the intraembryonic portion of the allantois and the vital intestinal duct. During further development, the endoderm gives rise to the following: the epithelial lining of the respiratory tract, the parenchyma of the thyroid, parathyroid, liver, and pancreas, the reticular stroma of the tonsil and thymus, the epithelial lining of the urinary bladder, and the urethra is also endodermal derivative. The epithelial lining of the tympanic cavity and the auditory tube is also endodermal derivative. At the cephalic end, the foregut, ectodermal and endodermal membrane or the oropharyngeal membrane separates the stomodium. This deep depression is the future mouth or the stomodium. It separates the stomodium, the primitive oral cavity, which is derived from ectoderm, from the pharynx, a part of the foregut, which will be covered later. In the fourth week, the oropharyngeal membrane ruptures, establishing an open communication between the oral cavity and the primitive gut. So at this time, the embryo swallows amniotic fluid. The hindgut also terminates temporarily at an ectodermal endodermal junction, that is the cloacal membrane. The membrane separates the upper part of the anal canal. This is proctodium actually, which will form uh, the anal canal. This membrane separates the upper part of the anal canal, which is the derivative of the cloaca, from the lower part or the ectodermal part of the anal canal. The membrane breaks down in the seventh week to create an opening of the anus. So at seventh week only, the uh, amniotic fluid passes from the oral cavity to the anal aperture. Another important result of the cephalocaudal growth and the lateral folding is the partial incorporation of the allantois into the body of the embryo, where it comes out from the cloaca. The distal part of the allantois remains as connecting stalk. As a result of cephalocaudal and lateral folding of the embryo, a portion of the endoderm derived from gastrulation is incorporated in the embryo to form the primitive gut tube. The yolk sac and the allantois remain outside the embryo. Due to formation of the cardiac bulge, the depression between the head end and the chest becomes very deep to form the stomodium, uh, at the base of which lies the buccopharyngeal membrane. Similarly, a depression develops between the attachment of the umbilical cord and the tail fold, at the base of which the proctodial membrane lies, and this depression forms the lower part of the anal canal. In the cephalic and caudal parts of the embryo, the primitive gut forms blind-ended tube, the foregut tube, and uh, the blind-ended tube caudally is the hindgut tube. The middle part remains as midgut tube, which is temporarily connected with the yolk sac through the vitalointestinal duct. The foregut extends from the oropharyngeal membrane up to the uh, liver bud as far as the liver outgrowth. The midgut begins caudal to the liver bud and extends up to the junction of the right two-third and left one-third of the transverse colon in adult, which is not demarcated in the fetus. The hindgut extends from the left one-third of the transverse colon to the chlorical membrane. Endoderm forms the epithelial lining of the digestive tract and gives rise to specific cells. This lining of the uh, stomach, duodenum, or other parts, the glands, the hepatocytes, exocrine and endocrine cells of the pancreas. The stroma of the glands is a derivative from the visceral mesoderm. Muscle, connective tissue, and the peritoneal component of the gut is also a derivative from the visceral mesoderm or the splagnopleuric mesoderm. When the embryo is approximately 4 weeks old, the respiratory diverticulum appears at the ventral wall of the foregut. The tracheoesophageal septum gradually partitions the diverticulum from the dorsal part of the foregut, and any abnormality of this partition will give rise to tracheoesophageal fistula. In this manner, the foregut divides into a ventral portion, the respiratory diverticulum, and a dorsal portion which forms the esophagus. At first, the esophagus is short, but when it descends with the heart and lungs, it lengthens rapidly. The muscular coat of which is formed by surrounding visceral mesenchyme. Uh, it is striated in the upper two-thirds and innervated by the vagus. The muscle coat in the smooth muscle in the lower part is smooth muscle, and it is innervated by the splagning nerve. The stomach begins its development from the foregut at 4th week as a fusiform dilatation in close proximity with the respiratory diverticulum in the primitive thoracic region. Growth to lengthen the esophageal region is essential for position of the stomach in the abdominal cavity below the diaphragm. If this growth does not occur, a diaphragmatic hernia results in with the stomach remaining in the thoracic cavity and compressing the lung. During following weeks, after lengthening of the esophageal region, the foregut, uh, the appearance and position of the stomach change greatly as a result of differential rates of growth of various regions of the wall. The greater and lesser curvatures are formed. The midgut. In the fifth week embryo, the midgut is suspended from the dorsal abdominal wall by a short mesentery, and it communicates with the yolk sac by the way of vitalointestinal duct. In the adult, the midgut begins immediately dorsal to the entry, distal to the entrance of the bile duct in the duodenum, and terminates at the junction of the proximal two-third and the distal one-third of the transverse colon. Over the entire length, the midgut is supplied by the superior mesenteric artery, which will be seen in the next slide. The development of the midgut is characterized by rapid elongation of the uh, the gut and its mesentery, resulting in the formation of the primary intestinal loop. At its apex, the loop remains open in connection with the yolk sac. Um, the cephalic limb of the loop, this is the cephalic limb, develops the distal part of the duodenum beyond the opening of the hepatic duct. Then it forms the jejunum, ileum, the maximum part of the ileum. The caudal limb comes to form the lower portion of the ileum, cecum, the appendix, the ascending colon, and the proximal two-third of the transverse colon. The hindgut forms uh, the remaining portion of the transverse colon, descending colon, sigmoid colon, rectum, and the upper part of the anal canal. The hepatic bud, it is an endodermal cord which penetrates the mesenchyme of the septum transversum. The septum transversum also contributes in the formation of the diaphragm. The pancreatic bud uh, develops from the endoderm into the dorsal mesentery. This is the sagittal midline section of the embryo at 5th or 6th week of development. The embryo during 6th week of development showing the uh, blood supply of the different segments of the gut, the vitelline arteries. There are multiple vitelline arteries coming from the dorsal aorta. Only three vitelline arteries remains. One is the celiac artery, which supplies the esophagus, stomach, and the proximal part of the duodenum. The middle one, which forms the superior mesenteric artery, it supplies the midgut region up to the right two-thirds of the transverse colon. The inferior mesenteric artery supplies rest of the transverse colon and the hindgut. The stomach is attached to the dorsal body wall by the dorsal mesogastrium and to the ventral wall by the ventral mesogastrium, which is part of the septum transversum. As the liver grows inside this region, the mesoderm forming the ventral mesogastrium becomes thinner and forms two parts of the ventral mesentery. The lesser omentum connecting the stomach with the liver. This is the lesser omentum, and the falciform ligament connecting the liver to the ventral body wall. The spleen becomes connected to the posterior body wall in the region of the left kidney by a linorenal ligament, which is a part or derivative of the dorsal mesogastrium. Due to the enormous growth of the liver and the proximal limb of the Yolk gut, there is herniation of the gut tube into the umbilical cord during the fifth week of uh, uh, development. Then here there is 90-degree development during herniation, and up to 10th week there is another 180-degree anti-clockwise rotation of the Yolk gut. Retraction of the gut occurs during the 10th week. During retraction, the remaining 180-degree anti-clockwise rotation occurs. The first portion of the gut to re-enter in the abdominal cavity is the jejunum and the ileum to the left side of the abdominal cavity, and the remaining portion or the colonic portion enters into the right side of the abdominal cavity and settles there. During 10th week, the herniated intestinal loops begin to return the abdominal cavity. Rotation occurs during herniation around about 90-degree anticlockwise, as well as during return of the intestinal loop remaining 180-degree. The hindgut gives rise to distal portion of the transverse colon, the descending colon, the sigmoid, the rectum, and the upper portion of the anal canal. The endoderm of the hindgut also forms the internal lining of the urinary bladder and urethra. The terminal portion of the hindgut enters into the posterior region of the cloaca, the primitive anorectal canal. The allantois enters uh, into the urogenital sinus. The cloaca itself is an endoderm-lined cavity covered at its ventral boundary by surface ectoderm, that is the cloacal endoderm. The boundary between the endoderm and the ectoderm forms the chlorical membrane. A layer of mesoderm, the Eurorectal septum, develops from the lower part of the mesoderm of the umbilical region, separates the region between the allantois and the hindgut. The septum is a derivative or is derived as a wedge-shaped mesoderm between the allantois and the hindgut. As the embryo grows, the caudal folding continues. The tip of the Eurorectal septum, which comes close to the chlorical membrane. At the end of the 7th week, the chlorical membrane ruptures. Actually, the chlorical membrane is divided by the Eurorectal septum into anterior part, that is the urogenital membrane, and the posterior part, the anal membrane, and the tip of the Eurorectal septum forms the perineal body, thus dividing the cloaca anteriorly to urogenital sinus, which gives rise to urinary bladder, urethra, and the posterior part, which gives rise to the anal canal and the rectum. The Upper part, two-thirds of the anal canal is derived from the endoderm of the hindgut. The lower one-third is derived from the ectoderm around the proctoderm. Ectoderm in this region of the proctoderm on the surface part of the cloaca proliferates and invagenates to create the anal pit. Subsequently, degeneration of the chlorical membrane, now it is called the anal membrane, establishes the continuity between the upper and lower parts of the anal canal. Because of the caudal part of the anal canal originates from the ectoderm, it is supplied by the inferior rectal arteries, the branches of the internal pudendal arteries. However, the cranial part of the anal canal originates from endoderm and therefore it is supplied by the superior rectal artery, a continuation of the inferior mesenteric artery, the artery of the hindgut. The junction between the endoderm and the ectodermal region of the anal canal is delineated by the pectinid line just below the anal columns. At this line, the epithelium changes from the columnar in the upper part to stratified squamous in the lower part. The liver primordium appears in the middle of the third week as an outgrowth of the endodermal epithelium at the distal end of the foregut. This outgrowth, the hepatic diverticulum or the liver bud, uh, it grows rapidly into the and the cells proliferate that penetrate the septum transversum, that is the mesodermal plate between the pericardial cavity and the body stalk. Whereas the hepatic cell continue to penetrate the septum, the connection between the hepatic diverticulum and the foregut narrows, forming the bile duct. A small ventral outgrowth forms the gallbladder and the cystic duct. During further development, the epithelial liver cords intermingle with the branches of the vitelline and umbilical veins which forms the hepatic sinusoids. Liver cords differentiates into parenchyma or the hepatocytes and the lining epithelium of the bile ducts. The Kupffer cells and the connective tissue are derivative of the mesoderm of the septum transversum. Development of the pancreas. The pancreas formed by two buds, the ventral originating from the endodermal lining of the duodenum, whereas the dorsal pancreatic bud uh, form the endodermal lining. It originates from the endodermal lining of the duodenum. Uh, the dorsal pancreatic duct grows in the dorsal mesentery. The ventral pancreatic duct grows close to the bile duct. When the duodenum rotates to the right and becomes C-shaped, the ventral pancreatic duct moves dorsally in the manner similar to the shifting of the entrance of the bile duct. Finally, the ventral pancreatic duct comes to lie immediately below and behind the dorsal pancreatic duct. Later, the parenchyma and the duct system of the dorsal and ventral pancreatic buds fuse. The ventral bud forms the uncinate process and inferior part of the head of the pancreas. The remaining part of the gland is derivative from the dorsal bud. The main pancreatic duct is formed by the distal part of the pancreatic duct and the entire ventral pancreatic duct. The proximal part of the dorsal pancreatic duct, the proximal part of the dorsal pancreatic duct is either obliterated or it persists as a small channel, the accessory pancreatic duct or duct of Santorini. The main pancreatic duct, together with the bile duct, enters the duodenum at the site of the major papilla. The um, the the entrance of the accessory pancreatic duct forms the minor papilla. In about 10% cases, the duct system fails to fuse and the original double pancreatic duct remains as pancreas division. In the third month of the fetal life, pancreatic islets develop from the parenchymatous pancreatic tissues. So the islets are derivatives of the endoderm, and the insulin secretion begins approximately around fifth month. The ventral mesoderm surrounding the pancreatic bud forms the pancreatic connective tissue.